Hello everyone, I hope you are enjoying this first ever virtual edition of Festival of Plants at Cambridge University Botanic Garden. I'm Gabriela Doria, a recent alumna from the Department of Plant Science and Wolfson College, and I'd like to talk to you about the imperceptible texture of petals and its role in pollination in wild tobaccos. While at Cambridge, I had the honor to participate in several versions of Festival of Plants at the gardens. And I'm very happy to be able to participate again this year from my lockdown in Bogota, Colombia. It was precisely in Colombia, one of the most biodiverse countries in the world, where I became fascinated by the beauty and diversity of plants. With more than 300,000 species, flowering plants are the most diverse group of plants on Earth. This diversity is mostly due to the complex interactions and long evolutionary history between flowers and their animal pollinators. Flowers have evolved in multiple ways to facilitate the interaction with their pollinators. We can perceive with our human senses some floral features such as color or smell. Here, for example, we have the famous titanarum with this red flesh petal-like structure and putrid scent to attract carrion flies for pollination. And here we have the South African daisy Corteria diffusa with this complex black spot mimicking female flies to deceive the males for pollination. Some other features are imperceptible to us, but also play important roles in the interaction between flowers and their pollinators. Here we have the narrow ridges on the petals of Hibiscus trionum that produce iridescence and can be detected by pollinating bees. In my research, I focus on one of these imperceptible floral traits. The cells on the surface of the petal can be of different shapes. They can be conical and extend out the surface of the petal, or they can be more or less flat. Because of their optical properties, conical cells enhance petal coloration. Here, for example, we have the wild type of the snapdragon Antirrhinum majus with conical cells and a deep dark pink coloration. In contrast, this mutant with non-conical cells, but the same amount of pigment background has a paler coloration. Conical cells have also been demonstrated to provide grip, which is important for the small pollinators such as bees. I studied petal cell shape in the genus Nicotiana, a group of about 75 species in the nightshade family from the Americas, Australia, and Southwest Africa. Specifically, I focus on sister species of Nicotianas that have contrasting characteristics of petal cell shape. One has conical cells, and her sister has non-conical cells. By comparing these closely related species, I was hoping to detect the mechanisms involved in defining petal cell shape. I focused on molecular mechanisms potentially involved in the development of petal cells. From previous work, we know that this group of genes is involved in the development of cell projections in other plants. For example, in the growth of cotton fibers. By comparing the gene sequences between the sister species of Nicotiana, I was hoping to find clues on how these genes are involved in defining petal cell shape in the genus. I characterized and compared the sequences of the sister species, and I found that although the sequences in general are very similar, there are differences in some genes between sister species with contrasting characteristics. But are these differences enough to explain the contrasting cell shapes? One way to test this is by expressing either version of the gene coming from the conical or coming from the non-conical species independently in a plant that originally doesn't have it. If the sequence differences were solely responsible for the contrasting petal cell shape, we will expect that only the plants expressing the version of the gene coming from the conical cell species will produce cell projections. However, this is not the case. Both sets of plants, those expressing the gene from the conical cell species and those from the non-conical cell species, were able to produce striking projections such as hairs and conical cells that were not present in the wild type. So these differences in the gene sequences are not explaining the contrasting cell shapes. Alternatively, it might be that differential expression of the genes along development might be involved in the contrasting petal cell shape. Here we can visualize differences in expression of these genes that is mostly absent during all stages of development of the non-conical cell species, while it's present in the early development of the conical cell one. It might be that the differential expression of one or more genes might be directing contrasting cell shape in this species of Nicotiana. To test the role of petal cell shape in pollination, I ran a series of pollination behavior experiments with bumblebees. I used artificial flowers mimicking the combination of features present in the sister species of Nicotiana. I used a high-resolution resin and pigment combinations to mimic the texture and color of the actual flowers. Here we can see a bumblebee visiting a red flower with non-conical cell surface, struggling to feed from it. And here we have the same bumblebee landing and feeding peacefully on a conical cell flower. Differential conditioning experiments showed that the bumblebees could learn to discriminate between artificial flowers loaded either with punishment or reward that only differ in texture but were otherwise identical. But why should we care about this pollination business? This map shows the percentage of GDP dependent on pollination by country. Here is my country, Colombia, where the number reaches 15%. I invite you to pause and look for your own country. Moreover, in a world where biodiversity is under high risk, understanding plant-pollinator interactions provides us with tools to contribute to the conservation of biodiversity of plants and animals. It also allows us to engage with plant breeders to optimize pollinator attraction and ensure crop security. I'd like to thank the support for my research, my supervisor, Professor Beverly Glover, all members of the Glover Lab, the organizers of Festival of Plants, and you for watching. If you had any questions, please feel free to reach out and don't forget to watch my movie. Be safe and enjoy the rest of the festival.